Today we will start with the same unit 1, concept of active site and enzyme substrate complex. As I discussed in the previous lecture also, that enzymes are made up of proteins and all proteins are not enzymes. This is the primary functional of any enzymes. But enzyme work as a biocatalyst. For that, it require activation site. And how this active site can work with the substrate and form ES complex as well as product formations. So the concept of active site and enzyme substrate ES complex is a fundamental to understand the mechanism of enzyme catalysis. So what is the mechanism? Mechanism of enzyme catalyst you can understand only with the fundamental understanding of enzyme substrate complex. Enzyme plus substrate that will form ES complex. And this ES complex, as I say, many times in the examination they are asking this statement, elaborate that statement that enzyme are a biocatalyst and it speed up the reaction. So you have to write like that. They are typically protein and they have a specific three-dimensional structure. They have a specific three-dimensional structure which include a region which is called as active site. And these active site, if characteristic of active site is are there, is a small, well-defined region on the surface of the enzyme where the substrate molecules bind and undergo a chemical reactions. So I am repeating what is the active site. Active site is a typically you can say the protein that has a three-dimensional structures as well as it is a region at which a well-defined region at which the surface on the enzyme it binds with the substrate molecules and thereby it undergo chemical reactions. So there are two theories involved for these enzyme substrate complex. One is lock and key models or another induced fit model. That already been taught to you in the previous lecture, even though I am repeating here. In lock and key model, the active site has a specific shape and that complement the shape of the substrate molecules, that allowing them to fit together like as lock and key fit together, and it will show lock and key model. These models suggest that the active site is rigid. There is no change in that and does not change its conformation upon substrate binding. However, now another models. Induced fit model. What it says? It proposed that both the enzyme and substrate, enzyme as well as substrate, that undergo conformational changing on upon binding. This model, the active site is flexible and can adjust its shape. Active sites is flexible and adjusts its shape to accommodate a particular substrate molecules. And the binding of substrate induce changes in the enzyme structure. This binding, flexibility of active site on the enzyme surface. So if this is flexible, then the flexible with the shape of the enzymes or shape of the substrate, then what will happen? It will change. So enzyme structure can also change in this fit and leading to them optimal fit between the two molecules. The formation of enzyme substrate complex occurs when substrate binds to active site of the enzyme. This complex is stabilized by the various bonding system. First is non-covalent interaction such as hydrogen bonding, electrostatic interaction, van der Waals forces, or you can say hydrophobic interaction. So these interactions ensure the substrate molecules are held in a precise orientation for the catalytic reaction to occur. And that will only happen in the induced feed model, where you will find the active site is flexible. Whereas in lock and key models, each key is specific to each lock. Okay, so that will not shape is specific and complementary to the shape of substrate molecule. This is the major difference between these active sites, concept of active sites, and models in which enzyme substrate complex is formed. So series of chemical reaction that is happening with the active site and substrate, and it is called as catalytic cycle. During this cycle, the enzyme facilitates the conversion of substrate molecules into a product by, now remember this word, by lowering the activation energy required for the reaction to proceed. Lowering the activation energy delta G0. Okay. The active site provide a specific environment. What type of environment? That promote a specific chemical reaction by providing a functional group or a metal ion or a cofactor to participate in the catalytic process. So the active specificity is crucial for enzyme functions. Enzymes are highly selective, meaning they can catalyze the specific reaction with the specific substrate. So specificity is determined by the shape, charge distribution, chemical properties of the active sites. And active sites, the amino acid residue play a vital role in substrate binding and catalysis. Active site amino acid residue play a role in a substrate binding and catalysis. So you can say that different enzymes have a different amino acid sequences in their active sites. Am I right? So that can also be asked in the examination. Why? Why different enzymes have a different amino acid sequencing in their active site, which gives them a unique properties and allowing them to catalyze specific reactions. Now, how these can applied in the various ways. So, understanding this, our overall active sites and enzyme substrate complex, it is very essential to understand the enzyme kinetics or enzyme inhibition, or you can say nowadays it is a drug design formulation through these enzyme modifications. So by elucidating the structure and function of the active site, any scientist or biologist can develop a strategy to modulate enzyme activity, and for therapeutic purpose, you can or design inhibitors that selectivity targets specific enzyme involved in the disease process. So this is the concluding remarks of these. Active site has a specific region. Enzyme substrate bind molecules undergo chemical reaction. Then the formation of enzyme substrate complex involve complementary interaction between active site and substrate molecules. It is stabilized by the non-covalent interaction, whereas catalytic convert to substrate into product. And the active site specificity is crucial for enzyme functions, and it determines the ability to catalytic functions. So concept of active site mapping that I will teach you later on.